Okay. All right, welcome to the Evolution Vanderbilt Seminar Series. We are going to wait a few minutes as all of the attendees join us. So we will be meeting at this time every Wednesday with a few exceptions. So next week we will not have a speaker. We would instead like to encourage you to attend Dr. Ann Tate's talk, which will be Monday at 4 p.m. as part of the Biological Sciences Seminar Series. Also, I'd like to allow Ann Hatmaker to make an announcement about the Evolution Student and Trainees Group.
Hi, everyone. I have two related announcements. So the first one is that there is an Evolution at Vanderbilt organizations for students and trainees, which is called EVOST. And if you would like to join this is for students, postdocs, you can email Abby Labella, and I should be able to put her email in the chat after I finish speaking. And then related to that, this semester, to kind of encourage community building in such a virtual environment, we are hoping to host a book club that will be for everyone involved in the Evolutionary Studies initiative, but primarily the books will be chosen and that kind of thing through the EVOST platform. And so it'll be kind of student-led and trainee-led. And so again, if you would like to get on the list of people who receives a Google form to be able to choose the book that we will be reading, this should not be a major time commitment, then I will put that information in the chat as well. Thank you.
Great, thank you so much. Okay, so without further ado, I'm going to hand it over to Dr. Malik Patel to introduce our first speaker for the Evolution at Vanderbilt Seminar Series virtual edition.
All right, thank you, Larissa. So it's my great pleasure to introduce Harmit. It's a special privilege because Harmit was actually my postdoctoral advisor. I'll spare you all the stories. So Harmit did his bachelor's in India in chemical engineering, but he said that he always had sort of a liking for biology, and the more he did chemical engineering, the more he realized that he wanted to do biology. And so he came to US to do his PhD at University of Rochester in New York. I imagine it was quite a shock to go from Mumbai, India to Rochester, New York, where he worked with Thomas Eichbusch and did some phenomenal work. And then he did his postdoctoral work with Steve Henikoff at Fred Hutchinson Cancer Research Center in Seattle. So Harmit has done remarkably well. He has won many accolades. I'll just mention a few of them here. So he is a Sloan Fellow. He was a Pew Scholar, which I believe he declined. He was a Searle Scholar. He won the NSF Career Award. He was also named the HHMI Early Career Scientist, and that's when actually I joined his lab. And then subsequently, he became a HHMI investigator in 2013. More recently, he became a member of the US National Academy of Sciences, and he's currently serving a 2-year term as president for the Society for Molecular Biology and Evolution. Harmit has published well over 100 papers. And so clearly there's no doubt that he's an amazing scientist. I had the opportunity to see that firsthand. But having been advised by Harmit and having worked so closely with him, I thought I'm rather in a unique position to tell a little bit about sort of a personal side of Harmit. So I think besides the scientific track record, what I think is remarkable about Harmit's lab is the fact that 11 postdocs from his lab have gone on to have their own academic positions at academic institutes. And this is a track record in the last about 16, 17 years since he's had his lab. As far as I know, this is one of the most remarkable sort of track records of successful mentoring of postdocs. And I think this is perhaps no surprise given how the kind of person Harmit is. You know, I think it's only in retrospect I realized sort of how rare sort of his talents are. So, you know, in order to have such amazing sort of a pool of individuals go on to have their own successful research programs, it takes, I think, a lot of generosity, I think, on part of Harmit, right, to allow people in his lab to really have the intellectual freedom to develop their own programs, and then actually to allow them to take those research programs with them. So this is something that, again, I think is quite rare in academia. And again, I'll just mention now that I have been a PI myself, I've come to recognize that while science is hard, mentoring is really, really hard. And I appreciate sort of the skills that Harmit has in this all the more. And I'll just end by mentioning something that Harmit has mentioned in a Q&A that he did for Current Biology, I believe just earlier this year. And so the interviewer asked him, you know, what do you see as your scientific legacy? And it's no surprise that Harmit said that, well, that's a no-brainer. You know, it's not really the science I've done, but really the people I've trained and the success that people have had in their own fields when they left my lab. You know, that's what he identifies as being his scientific legacy. And I think I can speak from first-hand experience that this is really Harmit speaking from his heart. So Harmit, it's a great pleasure to have you. And without further ado, we look forward to your seminar. Thank you.
Well, thank you very much, Malik, for those kind words. I think Larissa needs to make me the host so that I can share my screen. Sorry for the technical things. We just need to make sure that everything is done correctly so this can be recorded as well. So ready when you are, Larissa.
I suspect that she's working on it.
Yes, I think she's, I just want to make sure.
There we go. Okay. Malik, can you just turn your video off? Perfect. All right. Now I will go ahead and make you host.
Okay, thank you.
There we go. You should, there we go.
I think everybody can see my screen, hopefully. Let me know if you cannot. So it's a real pleasure to be speaking to you guys today. I must confess I'm a little bit disappointed to not be visiting you in person because I've tried to make it a point to visit all of my alumni labs and see how they're doing and thriving in their environments. And I'd also heard a lot about those hot chicken, Nashville chicken items from Seth Bordenstein, which I guess I will have to wait for like a parcel to arrive at my home. But I'll instead try to do my best to try to tell you about one aspect of the science in our lab, which is about how host-virus interactions can play out. And this is actually work that was primarily led by two former graduate students, Patrick Mitchell and Rosanna Colon-Tillier, and has been now actually taken on by a current postdoc in the lab, Jeanette Tenthore. Okay, so I'm really interested in classes of problems that could be kind of summarized quite nicely as the Red Queen hypothesis. The Red Queen hypothesis was actually pioneered by Lee Van Valen and his colleagues, primarily driven out of kind of ecological considerations, where they talked about how they could actually very accurately predict the population size of prey populations based on what was actually happening with predator populations in preceding years. And you had almost this precise cyclical nature of expansions and contractions of prey and predator populations because they were so intimately interacting with each other in fitness space. I'm interested in this class of problems where you basically don't just have to worry about adaptation to abiotic environments, but also have to worry about adaptation within genomes or between different entities that are vying for the same fitness space, where it's actually not possible for both entities to coexist in a kind of kumbaya equilibrium. They're basically both battling for supremacy in this fitness continuum. So I'm really interested in applying this really important insight to molecular problems, and it's really easy for me to translate this prey-predator interaction into that between hosts and viruses, particularly pathogenic viruses, where we basically have a situation where host proteins are constantly evolving to recognize and antagonize and extinguish viral infections, which poses an enormous amount of Darwinian pressure on viral populations to evolve new states such that they are no longer recognized and antagonized by the host protein, which now puts pressure back on the host population to now evolve a new variant of the antiviral protein that can reestablish its antagonism of the viral protein. The number of arrows are here to sort of remind you that this is an inherently asymmetric arms race, and we'll talk a little bit about the nature of this asymmetry in this seminar. But it's asymmetric for more reasons that you might actually suspect. It's not just asymmetric because viruses can evolve much more rapidly and be subject to much larger effective population sizes compared to host proteins, which are germline encoded and therefore are going to be evolving much more slowly. But actually, it's not just enough for a virus to survive one particular antiviral protein, but it has to survive actually a whole battery of antiviral proteins. And for the host to win, it actually only needs to win one of these interactions. Whereas for any given virus, it really needs to run the entire gauntlet of these interactions in order to be successful. But of course, on the host side, it's not that it's actually only contending with one virus at a time. It's also contending with many different viruses that are basically challenging host proteins. So how hosts are able to actually mediate these kinds of polynomial interactions is something that's really interesting to us. But at its essence, this is an arms race that can be boiled down in a very reductionist way to an arms race between an antiviral protein and a viral protein. And what drives this sort of engine for evolution is the fact that one party is always losing this arms race. Either the virus or the host is on the losing side of this arms race. And therefore, there's always going to be an evolutionary advantage to be gained by innovation. And what people like me are interested in is looking at signatures of this innovation as trapped in the sequences of host proteins and viral proteins to try to reconstruct how these arms races played out over millions of years of evolution and try to understand the biochemical underpinnings of how these arms races played out. So as molecular evolutionists, we actually have some really important tools. One very simple tool that we've been able to use is just to quantify this innovation as it occurs in protein coding genes, because all mutations in these genes can be classified as those that will not alter the amino acid being encoded, so-called silent or synonymous changes, and these can be normalized as DS, versus those that will instantly alter the amino acid being encoded, and those are non-synonymous or replacement changes normalized as DN. Now, we make the simplifying assumption that as far as natural selection is concerned, it cannot actually see these synonymous changes because it has not affected the final protein product. Now, this is clearly a false assumption. Those of you who are bacteriologists or yeast biologists know this quite well, where synonymous changes can actually profoundly impact rates of protein translation and even protein stability. But as a first order approximation, this works really well, where we can use the accumulation of silent or synonymous changes as a rough molecular clock of how much time has elapsed since the divergence of these genes, and use the incidence or retention of these non-synonymous changes as an imprint of how natural selection has acted on this part of the protein coding gene. So for instance, if you are looking at a protein coding alignment in which your eye is drawn towards those residues that are completely unchanged despite millions of years of divergence, that's an indication that DN is practically zero in spite of the fact that DS is quite high. That's because selection has acted to purify the population of these presumed deleterious amino acid changes when they occurred in the course of evolution. In other words, as molecular biologists, we use high evolutionary conservation to make predictions about functional importance. And that's actually the basis for reverse genetics, and that's absolutely true. But what we sometimes make is the false assumption that parts of the protein that are not very highly conserved must not be very important for function. In fact, my entire lab is interested in classes of genes and classes of protein domains in which the rate of apparent amino acid replacement actually exceeds that what we see at the synonymous or silent site. And that's not happening because these silent sites are suddenly much more important. That's actually happening because selection has accelerated the rate of fixation of these amino acid replacements because they were beneficial in the context that they occurred. So just like high evolutionary conservation is a proxy for functional importance, we use this constant innovation as a proxy for functional importance. But this is a very specific type of innovation that is potentially really informative about arms races. One very simple sort of cartoon diagram to look at the difference between purifying selection and positive selection is to consider a protein-protein interaction interface, let's say between two host proteins that are participating in a signaling cascade. Now, over millions of years of evolution, we expect that there will be mutations that affect one partner or the other partner that affects the interaction interface between these. But since I told you that the interaction between these proteins is really important for the host, any mutation in these interaction domains is going to be purged out as deleterious by natural selection. So you can actually come back millions of years later and find that purifying selection has largely maintained the interaction interface between these proteins. In contrast, in an arms race-like scenario, we again have a protein-protein interaction interface between a host protein and a viral protein, but now it's actually in the best interest of the host to maintain interaction, but in the best interest of the virus to actually evade interaction. As a result, this system will actually bounce back and forth between binding and evasion of binding as both parties try to either establish or escape this kind of binding interaction. So this is again a protein-protein interaction interface which should evolve the same as this, except this one is constantly rapidly evolving because of the disparate interests of the host and the viral proteins. Even from this cartoon example, you'll notice that actually we do not expect these changes in the protein-protein interaction domain to be randomly distributed. We expect them to be concentrated on those domains or residues that maximally affect the binding affinity between host and viral proteins. So armed with nothing more than this kind of simple concept, we sort of can ask, what are the types of genes that are subject to this kind of positive selection or disparate interaction? And we can do that quite simply because DNDS can be applied to all protein coding genes in the genome. So we can compare, for instance, all human genes to chimpanzee versus rhesus genes. And again, we see that there is an overwhelming signature of purifying selection, which means on average, most of the amino acid changes that have taken place between the rhesus human divergence have actually been purged out by selection. But nonetheless, there is a subset of genes which have evolved under very strong positive selection, and unsurprisingly, immunity genes are extremely overrepresented in this category. So this is a clear indication that immunity is subject to the type of rapid evolution or genetic innovation that I've been actually telling you about. So how do we use that signature? Just like what I told you, since we expect these changes to be maximally affected on the binding interaction surfaces of these proteins, we asked whether we can simply use positive selection or the signature of rapid evolution to identify host-virus interaction interfaces, even in the absence of any kind of biochemical knowledge of what these interaction interfaces are. So in other words, by asking where does positive selection occur, can we actually find out about how the interaction between these host and viral proteins have taken place over the course of evolution. So to introduce this concept, I'm actually going to tell you about studies we've done on one antiviral protein, which is a protein called MXA, stands for myxovirus protein A. It's an interferon-induced dynamin-like GTPase. There's a number of kind of terms here, which are not all important for the purpose of this talk, but I just want to remind you that interferon is the alarm that your body sends out when it detects some sort of pathogen. It's a pretty non-specific signal, which means you turn on a whole battery of genes, hoping that one of them will take care of the pathogen and attack. And MXA is, in fact, one of the most abundantly produced genes in response to this interferon signal. MXA is a distant relative of dynamin, which is important for endocytosis. Whereas dynamin participates exclusively in the cellular process of endocytosis, MXA has now specialized specifically for an antiviral role, but just like dynamin needs GTP hydrolysis in order to exert its antiviral function. So if you are a dynamin expert, this crystal structure of MXA is going to look a lot like dynamin, except it doesn't have the ability to interact with plasma membrane. It actually instead has the ability to interact with viral components. MXA was actually discovered because of a happy accident of mouse breeding. It turns out that most laboratory strains of mice are deficient for MXA, which means that if you expose them to even bird-adapted influenza, these mice are exquisitely susceptible to influenza infection. However, you can completely rescue this by just putting in either the human or the intact mouse version of MX, and you can do so even in a mouse that is otherwise debilitated for the interferon response, which means these mice do not have access to the hundreds of genes that are turned on with interferon. They only have access to one gene, and yet that gene is able to basically protect these mice against an otherwise lethal viral infection. Just sort of impressing upon you the importance of the genes like MXA for actually providing that initial block of viral infection that's necessary to allow the adaptive system to kind of catch up. So this was actually one of the first mammalian antiviral genes to be discovered, but more recently it's been realized that it just doesn't work against influenza-like viruses, but MXA actually can work against a whole panel of negative sense-strand RNA viruses, also positive sense-strand RNA viruses, as well as DNA viruses, which is actually a remarkably broad specificity for an antiviral protein. Typically, these proteins specialize for certain classes of viruses. So we became very interested in MXA because of this broad specificity, but also because in each case, MXA appears to recognize a completely unrelated component of the virus in each of these cases, which are totally non-homologous and unrelated. So it's not that MXA is going after one ubiquitous common component of these viruses. It's actually going after completely different components. And remember, these viruses can rapidly evolve. So these potentially components can also rapidly evolve. So how does MXA maintain this antiviral breadth while recognizing these multiple distinct viral targets is really kind of the motivation for this, not just to understand how immunity evolves, but also just as a protein engineering problem. I just want to give you a buyer beware here that I'm not going to actually answer the question, but all of the studies that I'm going to tell you about today were really motivated and are still motivated by this question. So one of the problems with this is that we actually did not know about MXA's interaction surface with any virus, including the NP protein or the nucleoprotein of influenza viruses, for which MXA is actually best known. So this is what prompted Patrick Mitchell, a former student in my lab, to undertake an evolutionary analysis where he sequenced MXA from a whole panel of simian primates. Luckily, today we don't actually need to do that because a lot of these simian primate genomes have been sequenced, but we were really interested in making sure that we had really good quality sequence so we can actually infer the action of natural selection on every amino acid of MXA. And unbeknownst to us, actually a different lab at University of Freiburg was actually attacking this problem, not from an evolutionary standpoint, but from a kind of virology standpoint. And once we realized that, Patrick and Corina from our collaborators lab actually joined forces to tackle this problem. So based on this, what Patrick discovered was that MXA actually is predominantly a slowly evolving protein, actually not very different from dynamin. So it's 660 amino acids long, and 85% of these amino acids are basically completely frozen in the course of primate evolution. So you might actually take this first pass view and conclude that MXA evolves under purifying selection and walk away. But because Patrick had sequenced all of these primates, he was able to slide a window one amino acid residue at a time and discover that in fact there were 12 amino acid positions which bore this clear signature of positive selection. And they were actually concentrated in specific sites in the crystal structure of MXA, which is a really good indication to us that these might be interaction interfaces because they're also hotspots for positive selection. In fact, the most interesting to us was actually this unstructured loop, which I've drawn in with my artist rendition because this is not part of the crystal structure. This is actually the biggest hotspot for positive selection. And in fact, is going to be the primary sort of focus of my story today, is to sort of try to understand what are the functional consequences of this rapid evolution. So armed with this signature that there has been positive selection of on MXA, Patrick wanted an assay. So we turned to a virus that our collaborators at the University of Freiburg had used quite a bit, in which this is a tegodovirus, which probably many of you have never heard on. It's actually a tick-borne virus that is quite lethal to rodents, especially in Europe. Just like influenza, though, it has the advantage that we can completely reconstitute the viral polymerase, and in an NP or nucleoprotein dependent fashion, which acts like a transcription factor to drive the expression of a reporter, in this case, luciferase. So by using a transfection control, we can get a pretty good readout of how much NP activity is occurring. And as we basically bring in different types of MXA, we can actually see how much they've affected the NP activity by how much this reporter activity is dropped. So it's a really nice way for us to actually assess viral fitness without necessarily having to do viral infections for all of our assays. So this is what Patrick already knew before he started his studies, which is that wild-type human MXA is actually really good at restricting tegodovirus in this replicon assay, as well as in infections. However, a catalytically dead version of human MXA completely loses this restriction and makes it almost like an empty vector control. What Patrick added to this was actually results from the rest of these simian primate MXAs that he had identified. And what we were really surprised by was like the fact that human MXA was actually the very best in terms of restricting tegodovirus. In fact, most other primates had really no measurable activity against tegodovirus whatsoever, in spite of the fact that these are all wild-type versions of MXA that actually work against other viruses. So it's not that we were unfortunate to get dead versions of MXA genes from these primates. It's just that these other primate MXAs have just not got the right specificity to actually restrict the tegodovirus. This is actually seen even more abundantly in a pairwise comparison between human MXA, which is very good, and African green monkey, which is very poor against tegodovirus. So what we can do is we can actually add increasing amounts of human MXA. And as you can see here, the more MXA you add, the lower and lower the viral fitness equivalent happens. Whereas at very high levels of expression, African green monkey MXA still does not have any measurable activity against tegodovirus. Keep in mind that these two proteins are more than 90% identical. So at this point, we basically have two MXA backbones which differ dramatically in their ability to restrict this particular virus. So biochemists in the audience are basically going to go, let's chop and dice and figure out what domains are responsible. But since we are evolutionary biologists, we like to pride ourselves on doing the minimum number of experiments to try to get to the answer. So we focused on signatures of positive selection to be our guide to do these kind of chimeric analysis. So what we basically did was we first took the human MXA backbone and replaced just the 5 amino acid residues in loop L4 that were under positive selection from African green monkey. Now it turns out that 2 of those residues are identical. So this is only a change of 3 amino acid residues. And you'll notice we've completely lost human restriction of tegodovirus. But the more dramatic kind of signal came when we add the 3 amino acids from human MXA into the African green monkey backbone and showed that that was actually sufficient to recover almost human-like protection against the tegodovirus. So what we've effectively done is we've not focused on the most conserved parts of MXA, but actually the least conserved parts of MXA. And a swap between these least conserved residues has allowed us to actually swap the specificities between these different viruses. Things got even more interesting because when Patrick did each of these residues one at a time, and now we've moved to infections because we have candidate loci, candidate residues, you can see that wild-type human MXA is really good at restricting tegodovirus infection, but a single residue change in loop L4 at residue 561 completely loses this restriction. Whereas wild-type African green monkey MXA, which has a valine, is very poor, a reciprocal change, bringing in a phenylalanine at position 561, completely rescues this. So I just want you to imagine, here's a single amino acid change that was driven by a single nucleotide change that's actually sufficient to completely change the stakes from the host losing to the host winning this arms race against a potentially pathogenic virus. This is why the selective coefficients associated with these changes when they occurred in evolution was so profound that they led to the very rapid fixation of these amino acid altering changes, which is one of the main reasons why we are able to identify these changes, even though we are averaging over 40 million years of primate evolution, which is mostly irrelevant in terms of Tagorevirus infection. Nonetheless, when they occurred, they left a clear imprint of their selective benefit. It turns out that Corina Pacina was actually doing the same experiments with influenza, basically premised on Patrick's result, and she found that the very same residue is also the key to restricting influenza. So human MXA is actually one of the best firewalls we have against bird influenza, which is one of the reasons more influenza variants don't jump into the human species. And yet, if you basically replace that single amino acid change, we lose most of that protection. Whereas African green monkey, which is not very protective, can be made more protective, again, by a single amino acid change. So I just sort of want to summarize what we've done here is we focused on residues under positive selection to track down specificity domains, and now basically specificity altering single residue changes. We took a little bit of a closer look at this particular residue at 561 and realized that we needed for this residue to be either a phenylalanine or a tyrosine, or to some extent a tryptophan, which are all hydrophobic aromatic residues. No other residue actually worked at that position, suggesting that there was a deep specificity about what needed to be present at these residues, presumably because this is where it actually interacts with a hydrophobic pocket in the NP protein of the Tagorevirus or the influenza nucleoproteins. So what's the biochemical consequence of this? Remember, I premised this on a binding affinity interaction. So what Patrick did was something kind of completely crazy, which is that he took the human L4 and actually put it on a mouse MX1 backbone. Now keep in mind that these MXAs are actually very rapidly evolving and now diverged by 80 million years of evolution. I already told you that mouse MX1 actually has very poor activity at protecting against Tagorevirus, and yet bringing in the loop L4 in a modular way was actually sufficient to give you significant gain of protection against Tagorevirus. And this gain of protection completely coincided with a gain of interaction avidity between the MXA protein and the Tagorevirus nucleoprotein. So you'll see here in these co-immunoprecipitation experiments that human MXA can very nicely co-IP with the nucleoprotein of Tagorevirus, whereas mouse MX1 has very poor ability to co-IP. And yet mouse MX1 with a human L4 swap, you can see, has recovered its avidity and also recovered its restriction ability against the Tagorevirus. So this entire arms race is being played and won and lost at the level of binding affinity. If you're able to bind the viral target, you're able to exert your antiviral action. Otherwise, you're not. So it might actually surprise you that single residue changes can have such a dramatic impact, given that, you know, nothing makes sense in that sense from a biochemical avidity standpoint. But one thing that I haven't actually told you is that just like dynamin, MXA acts as an oligomer, and the NP protein it binds is also an oligomer, which means that MXA sort of makes a concentric ring around the nucleoprotein, ribonucleoprotein complex in order to exert its antiviral action. So even small changes in the free energy of binding or at the monomer level basically translate to a dramatic change as you multiply that avidity over the course of the oligomer, which is why you have this almost switch-like state where a single residue change can give you such a dramatic difference in the outcome of this particular viral infection. So by focusing just on where positive selection occurred, this has actually turned out to be a really useful tool for us to identify not just the potential interaction interfaces, but also the susceptibility determinants that distinguish one from the other species version of the antiviral proteins. So we can actually graft these versions from our primate relatives onto the human protein to confer onto human proteins, often with single residue changes, the ability to actually restrict against viruses that we really truly care about. Now, what I'd like to emphasize here is that actually this is an entirely evolutionary principle. This does not actually rely on any components of virology or biochemistry, and yet this very simple principle has worked quite well for a whole panel of different viruses that work with completely different mechanisms, and the viruses themselves have a completely different life cycle. I also want to emphasize this particular virus that Malik actually worked on to look for escape from hepatitis C proteases in a protein called MAVS in primates. The reason that this is actually almost working so well universally is because despite the biology of these antiviral proteins being completely different, what they have in common is that the binding affinity with the viral proteins really dictates the ultimate outcome of the fate of this viral infection in these host proteins, which is why in all of these cases, we've been able to identify positive selection and identify functional changes because of single or double residue changes here. If you'd like to read a little bit more about this, there's a couple of reviews we've written. Here's one that I really like, which was written by a former postdoc, Matt Doherty, in Annual Reviews of Genetics, to get you a little bit more insight. So what we've done here, I don't want to leave you with the sense that loop L4 is the entire game in town, because what I've demonstrated is that loop L4 is really important to bind the nucleoproteins of influenza and influenza-like viruses. But we've also actually done studies in which we've shown that loop L4 sequence is completely irrelevant to binding other nucleoproteins from other RNA viruses, including VSV. In fact, we can make completely random changes in the loop L4 and not affect a nucleoprotein binding of VSV, which suggests that MXA has really used the Dynamin architecture almost like a Swiss Army knife, where each of these rapidly evolving domains is a potential interaction interface with a completely different class of viruses, where MXA is able to simultaneously battle each of these viruses, but by doing so by a completely specific kind of mechanism. So this is something where we can actually really use this evolutionary insight to really understand a little bit more about the biochemistry. Everything I've told you so far has been a retrospective view, where we've looked backwards in evolution to identify specificity domains. So when Rosanna, a new graduate student, joined the lab, she was really interested in whether we can translate this to a prospective view, which means can we use the same signature that we've identified from our evolutionary studies to actually use laboratory experiments to design or even obtain completely new specificities in existing antiviral proteins. So she was really interested in loop L4 because we had already kind of established that that's a really important determinant of influenza binding. And so she decided that if she focused just on loop L4, she could actually look at the combinatorial value of mutations in each of the 5 residues to ask what is the evolutionary constraint acting upon loop L4, but also what is the antiviral potential of loop L4. So even though Patrick had actually sequenced 25 different simian primates, that's still a very small fraction of the 20 to the 5th possibilities of loop L4, even keeping the rest of the human MXA backbone identical. So that's what Rosanna did. She used PCR mutagenesis to only mutate the positively selected residues in loop L4, obtain variants, and then test them in triplicate against Togotovirus replication. Because this PCR strategy cannot actually prevent stop codons from creeping in, we actually had a number of variants which had stop codons, which acted as a really good negative control. Also, a number of synonymous sites kind of crept in. So we had a number of versions of wild type MXA, which kind of defined the range of the restriction and the non-restriction activity. So based on that, she actually ordered each of the nearly 1,000 variants that she obtained from very low to very high restriction activity. These dotted lines represent the range of what wild type human MXA actually looks like. So everything below this dotted line is worse than human MXA. Everything above this this dotted line is actually statistically better than wild type human MXA. So these non-restrictors actually did not reveal anything super interesting, except that 95% of our variants were in this non-restrictor range, which is not like the most amazing news to get from your graduate student. 5% of our versions were either restrictors or what we call super restrictors. And when we took a look at these sequences, it was very apparent what mistake we had made, because all of these had one thing in common, which is that they actually had a phenylalanine, a tyrosine, or a tryptophan at these residue 561, which, if you recall, was exactly the same residue that Patrick had found was really important for Togotovirus restriction. So what we thought was actually a pairwise constraint turns out to be a universal constraint, where all the versions that have any activity need to have one of these 3 hydrophobic aromatic residues. But interestingly, that's actually not sufficient for Togotovirus restriction, because we have many versions that have the right residue, but are actually completely poor against Togotovirus. So residue 561 is necessary to be hydrophobic and aromatic, but that's not sufficient for Togotovirus restriction. So one of the most kind of profound contributions that I made to this project was actually to convince Rosanna to do the screen again, but this time fixed phenylalanine at residue 561 so that most of the variants that we obtained were likely to be in the restrictor range. And she was actually helped by a rotation student, Emily Shea, as part of this project. And indeed, luckily for me, this was actually a lot more successful because now more than 65% of the variants we obtained were as good or actually better than human MXA. What's really nice about this, though, is now we've got like a whole panel of versions that are significantly better than human MXA. I actually don't want to leave this slide without making the point that, again, despite having the right residue at 561, we still have about 35% of variants that are suffering some sort of negative epistasis from the other rapidly evolving residues in loop L4. We've not actually explored that very much because our focus is on how did we actually manage to improve upon human MXA's activity against tegodovirus, particularly because by now we have actually convinced ourselves that human MXA is actually the best mammalian MXA activity against tegodovirus that we've actually discovered. So what Rosanna did was she basically, again, lined up all of the super restrictors. And as we looked at that, we actually realized that actually some of the best super restrictors had an overabundance of Q at the very first position. Q is actually not that abundant in the genetic code. So we thought that, aha, that might be a clue that the first position, which we had allowed to mutate randomly, might be really important to actually have a Q. And indeed, that turned out to be the case because each of these super restrictors that we obtained were actually better than wild type MXA at every sort of level of expression, which is exactly what we want to see. So keep in mind that wild type human MXA is kind of our current gold standard for anti-tegodovirus activity. And in the laboratory, by just messing with the positively selected residues, we have versions that are actually significantly better than the best natural restrictor of tegodovirus. So how does this work? Well, each of these best super restrictors are actually better at avidity of binding to the nucleoprotein, exactly like what we had seen before. So once again, human MXA is actually quite decent at binding the nucleoprotein, but each of these super restrictors actually can bind much more significantly and much better. And that's actually the basis for this increased restriction. So going back to those Q residues, we actually mutate those Q residues back, reverted them back to the wild type human version, which was glycine. And in each case, we actually saw a drop of that super restriction, either back to the restrictive state or actually even worse than the restrictive state, suggesting that residue 540 indeed was really important to contribute to this higher than wild type restriction profile. So Rosanna actually made a single residue change at this position to all 20 possible amino acids and compared that to a catalytically dead version. So the first thing I want to point out is that putting a proline in a loop is predictably going to be bad. And indeed, we convinced ourselves that at this position, proline was as good as putting in a catalytically dead version. But actually, for the remainder of the residues, we had still a remarkably dynamic range. We're going from a tryptophan at that position to an alanine, which was clearly the best. We had a nearly 10-fold range of restriction differences between these variants that are only different at a single amino acid residue, with the wild type human version being smack dab in the middle. Now, it was really interesting to us to see that the three worst residues we could have at the first position, residue 540, were a tryptophan, a valine, or a threonine. The reason this was really interesting is because of our three very best restrictors, one of them was actually a valine and one of them was a threonine, which is like very unexpected given what Rosanna just found. So we inferred that if we were to take this valine residue and make that into an alanine, perhaps we could actually augment the super restricted activity even more and therefore get like a super duper restrictor version potentially. So just to sort of give you a sense of what we're looking for, here's the catalytically dead version. Here's wild type human MXA. This is the single residue change in that wild type backbone where a valine is much worse and an alanine is much better than wild type human MXA. So we wondered what would happen if we took a super restrictor that actually had a valine and converted that to an alanine. And just like most of my predictions, this turned out to be completely wrong because we took the super restrictor and when we converted that valine to an alanine, we completely lost the super restriction activity, which means that in fact, all of these residue changes, the grammar is very context specific, which is a highlight, of course, of epistasis. This is not a situation where we can pick the best residues at each of those positions and actually combine them. That is not going to be a successful strategy because these are actually interacting with each other. So far what I've told you is that we started with Patrick's observation that LUPL4 is a really important determinant of restriction against not just Tagorovirus, but also influenza virus. And in fact, one position, 561, was completely necessary to be one of these hydrophobic aromatics for both of these. And what we have now done is we basically allowed the rest of the other LUPL4 positions to mutate to get even more potent versions of Tagorovirus, which is probably a virus that most of you have never heard of before, but many of you have heard of influenza virus. So we wondered what would be the fate of these super restrictors against influenza virus. Now, there are three possibilities, of course. One is that it made no difference at all to influenza virus. That would be very disappointing. Possibility number two, which would be very exciting, is that in fact, these are also super restrictors of influenza virus. But the third possibility is that actually, as we get better and better against Tagorovirus, we might actually get worse and worse against influenza virus. And in fact, the third possibility is closest to what Rosanna found, whereas she basically tested each of these super restrictors. These are now rank ordered in terms of increasing Tagorovirus restriction. She found that most of them are actually worse than wild-type human MXA against influenza restriction. However, we did have 4 exceptions to this, where 4 out of the 24 super restrictors against Tagorovirus were not worse against influenza virus. So it's actually formally possible in this grammar of 5 residues to evolve new specificities without losing existing specificities, which is something that we're really kind of excited about. So what's actually going on here? Everything I've told you, of course, is entirely about this particular single loop, which is the hotspot for positive selection that we focused on. And this loop, I'll remind you, was unstructured, even though it was included in the protein in the crystal structure. So we wonder whether this is in fact biologically meaningful, because the fact that it was unstructured means that it did not just adopt one conformation, but in fact was very, very structurally flexible. And that might actually explain MXA's broad antiviral range against these orthomyxoviruses, because it is able to adopt multiple different structures to bind multiple different NP proteins. And what we may have done inadvertently in our super restrictor experiment is actually make slightly more preformed versions of LUPL4, which are of course much better against one type of binding to the NP protein, but lack the flexibility to adopt different conformations, allowing them to bind other NP proteins, which might actually explain this breadth specificity trade-off. And we're very interested in asking that. Another thing that we're very interested in asking is, can this breadth specificity trade-off also apply to other antiviral proteins? And that's kind of where Jeanette kind of entered the scene, where she was really interested in asking both the sort of biochemical underpinnings of this interaction, but also the how far can we extend this kind of paradigm to other kind of proteins. So I'm going to take a very brief segue to tell you a little bit about what Jeanette's been doing. And she's been actually focusing on a different antiviral protein called TRIM5, which is actually one of the most potent restriction mechanisms we have against HIV-like retroviruses. So when HIV enters the cell, it sheds its envelope at the cell surface and is basically now bounded, its genome is bounded by this protein capsid. And what TRIM5 does is basically it engages with this capsid really quickly and targets it for degradation, preventing it from undergoing the rest of its life cycle, and therefore basically blocking viral infection. TRIM5 was discovered because it had been known for a very long time that rhesus macaques cannot get infected with HIV-1, even if you actually actively infect them. And that's because each cell of their rhesus macaques encodes a version of TRIM5 that is highly potent at blocking HIV infection. We also encode a version of TRIM5, but our version is actually quite poor at restriction of HIV, whereas the rhesus version is really good. And now you can sort of see that most of these differences are because of positive selection, if you've been kind of keeping up. And we can actually explain most of the differences because of residue changes at one of these very rapidly evolving domains of TRIM5, which is the V1, which is the equivalent of the LUPL4 in MXA. What's really nice though is that we can actually graft, just like with MXA, rhesus-like activity onto human TRIM5 just by grafting in the V1 loop and actually get significant boost of activity just by a single residue change at one of these positions. So you can compare the top sequence and the bottom sequence, which is a 500 plus amino acid protein, but we've only mutated one of these residues motivated by positive selection and gotten almost a 15-fold increase in restriction range. So what Jeanette was actually puzzled by was that even though these are rapidly evolving positions and have changed over evolution, the mutational constraints still need to be explained though, because it was not that there were many different amino acid residues that were actually allowed at these positions. In fact, most of them have actually only toggled between 2 or 3 amino acid changes over the course of primate evolution. This led to the possibility that maybe helpful mutations that give you gain of function against viruses might be very rare in evolution. But of course, keep in mind that all of these sequences that we've obtained are from living primates. So maybe they've already undergone both mutation and natural selection. There's no way for us to decouple the effects of mutation and mutational bias from natural selection. So Jeanette decided that she's actually going to focus on this problem to assess what the shape of the mutational landscape is for human TRIM5 alpha, looking at all possible mutations of the V1 loop in the human TRIM5. So this is now a slightly different strategy than what Rosanna did, where Rosanna did combinatorial mutagenesis, whereas Jeanette is actually interested in only single mutation changes to allow her to see how do you walk up or down in this mutational landscape of human TRIM5 alpha against HIV. To ask the question, are winning solutions really rare or not? So to do this experiment, she needed to devise an assay. So of course, the deep mutational scanning part has actually been done quite nicely. We can do this with PCR mutagenesis to make single amino acid changes in each residue of V1, keeping the rest of the TRIM5 completely constant. And she was able to basically generate a library of cells, each expressing one version of this mutated TRIM5 variant. And what she did next was that she actually exposed the cell library to an HIV virus that actually was capable of expressing GFP upon integration. So any cells that lit up as green were cells that did not prevent HIV from going through its life cycle because they're now green, and therefore are likely to be encoding non-restrictor versions of TRIM5 alpha. All cells that are not green are either expressing restrictor versions or are actually were lucky enough to not be infected in the first place. So to actually keep out these not very interesting versions, we basically went through a second round of HIV infection to now again sort through cells that after 2 repeated rounds of infection remained non-green. And by comparing this final pool with the initial pool, we can basically actually look for versions of TRIM5 alpha that were restrictors versus the input library that went into the assay. What's really nice is that when she did this with 2 replicates, the replicates were like bang on, which means that in fact, what we were looking for was biologically meaningful. And when she looked at the sequence of these replicates, they made a lot of sense because as we look for the enrichment in the restriction pool, human TRIM5 and synonymous versions of human TRIM5, remember, human TRIM5 is not very good at restricting HIV, were actually all in the sort of tending towards non-restrictor pool. All stop codons were in the non-restrictor pool. But the surprising result was actually that we had many variants that were actually better than human TRIM5. So to quantify this enrichment score with meaningful viral restriction, Jeanette actually turned to this assay where she was able to infect with increasing viral dose to deduce the viral dose needed to infect cells at 10% frequency. You'll notice that if you have an empty vector, you basically can get to this 10% level with a very small viral dose. Because human TRIM5 alpha is a very poor restrictor, you actually need very little viral dose to get up to restrict human TRIM5 alpha. But the version that I already showed you with a single proline change, you needed a much higher viral dose. So we can use this ID10 assay to actually assess how much better is the TRIM5 variant that we are assessing relative to the empty vector control or to the wild type human TRIM5. And so what we did was we actually individually tested many of these versions across the entire range. And this was work that was mostly done by Candice Young, a research technician, and Afeez Soden, who was a summer student in our lab. And what the take-home message is that this beautifully matches our enrichment score. So the fold enrichment score beautifully matches what we see with fold restriction of individually tested variants against HIV. So even though this fold enrichment score comes from a pooled assay, it beautifully matches what we see with the HIV assay in individual scores. So the really important kind of take home is that 55% of variants improved HIV restriction. And in fact, there were some positions that really stood out for us. So keep in mind now that the bluer you are in this assay, the better you are in restricting HIV. And you'll notice that actually there were two positions in particular where human has an arginine at 332 and 335, and anything other than an arginine or a lysine is actually much better at restriction. So we are actually doubly unfortunate with having this diarginine here. Actually, if we had anything other than an arginine at either of these positions, we would be significantly better at restricting HIV. There was also another important insight that any position changed to an acidic residue, an aspartic acid or glutamic acid, gave you gain of function. And there were other things which I would encourage you to check out the preprint that's out there, where conserved positions, where we thought that maybe this position is really important for function, turns out it's actually not that important for function. We have no idea why it's constrained as a glycine, because anything other than a glycine appears to be kind of an improvement. So what we are basically discovering is that actually charge appears to be the predominant driver of HIV restriction of TRIM5 restriction of HIV. And we can explain most of the diagram that I just showed you just by a gain or loss of electrostatic charge, suggesting that actually this TRIM5 interaction with capsid might really be driven by this charge repulsion, which is an insight we would not have actually obtained by single residue changes. But what I want you to focus on is the fact that 50% of these missense mutations actually increase HIV restriction. And yet we have fixed these double arginines at this position. So this begs the question whether these double arginines are fixed for some other reasons, maybe some other HIV-like virus that was really needed to protect against this. So far, we've actually not arrived at that virus, because it actually turns out that human TRIM5 is really a poor restrictor of most HIV-related viruses. And in most cases, you're actually better off getting rid of that charged arginine to see gain of functional restriction. We actually have no cases where that proline is actually worse than the arginine in terms of restriction profile. So the hunt goes on for this mysterious virus which might have driven this very important change in human TRIM5. So the solution is that actually winning solutions are remarkably not rare at all. In fact, if we did a random walk in sequence space, 55% of the time, we would actually end up with a winning solution that was significantly better than the version that we actually have in the genome right now. So we wanted to ask the opposite question. Okay, Rhesus TRIM5, which is really good, it sits on one of these fitness peaks. How stable is its winning solution? Is it the case that Rhesus TRIM5 can actually tolerate a lot of changes? Or is it the case that Rhesus TRIM5 is like one of these black diamonds where it's actually very perilous that it's got this gain of function, and any kind of mutation is more likely to take it off this peak? So we wanted to ask using the same kind of principles, but this time, since we were starting off with Rhesus, which is very good, we actually focused on versions that turned green, which were basically non-restrictor versions of TRIM5 alpha that were not able to prevent HIV from infecting these cells. So now we are actually turning our assay from a restrictor enrichment to a non-restrictor enrichment. And I'll go really quickly here, because exactly like what we saw before, the replicates correlate beautifully. And exactly like we predicted, the nonsense mutations were highly enriched in a non-restrictor pool, whereas the wild-type versions of Rhesus TRIM5 were actually all great restrictors. But again, what I want you to notice is that 50% of the single mutations really did not significantly affect HIV restriction at all, which is remarkable, which means that actually Rhesus is actually quite resilient to single mutational changes. In fact, there were some patterns that we saw. For instance, again, positive charge is generally detrimental, and you're better off having a negative charge. But they were not like dramatic kind of things that we saw, except that this proline residue, again, appears to be important, perhaps because it provides an important kink in this V1 loop. So what I kind of want you to leave with this notion that, okay, for HIV, rhesus TRIM5 alpha is remarkably resilient to mutation. We tested against a completely different virus. This is entropic murine leukemia virus, which is a different class of retroviruses. Again, rhesus TRIM5 alpha is a very good restrictor of MLV. And again, we saw that 58% of single residue changes were actually made no real difference to the restriction profile. Similarly, human TRIM5 alpha, which is a dynamite restrictor of MLV, 92% of missense mutations had absolutely no impact on human restriction. So if you've been keeping track, what I just told you is that rhesus TRIM5 alpha, which is very good at HIV, I can mutate, I can make mutations in 51% of the time. I can make single residue mutations and not affect this activity. Against MLV, I can do the same thing, and 58% of the time I can make no difference. Human TRIM5 alpha is also remarkably resilient in terms of its ability to protect against MLV. What I haven't been able to tell you is how does human TRIM5 alpha do against HIV, because of course human TRIM5 alpha is not a good restrictor of HIV. But what we were able to do is actually do like a conversion where we converted human TRIM5 alpha to a potent restrictor of HIV by a single residue change at R332P, and then mutate the rest of the V1 loop. And once we did that, we actually found that this version was also highly resilient, which means that even though you can gain functionality against viruses very, very quickly, just with single amino acid changes, it's not the case that that functionality is easily lost by subsequent mutations. This is, of course, a very important insight because mutational resilience allows TRIM5 alpha to adapt rapidly in its V1 loop without actually constantly losing the ability to restrict the viruses that it was previously able to restrict. So it suggests that winning solutions are actually quite stable, much more so than we would have actually predicted at the onset of our experiment. Which means that even though there is a huge advantage to viruses in these arms races, because of the mutational resilience and the polynomial nature of the antiviral response, there are actually significant advantages on the host side as well, where its evolutionary landscape is actually not as constrained as we previously believed. So this is all I have to tell you about primate viruses today. I just want to leave you with the notion that this kind of insight actually traverses many types of arms races, including an arms race that I'm particularly excited about that I don't have the time to tell you about today, between phages and bacteria, where bacteria are now the host and use CRISPR-Cas systems to defend against phage. And phages use a whole variety of anti-CRISPR strategies in order to do that. And actually that's a project that was actually pioneered by Kevin Forsberg in my lab. And as Malik told you, this is a project that he completely initiated and will be taking with him onto the job market this fall. So if you guys have searches open, I would really encourage you to keep an eye on for both the evolution as well as the evolutionary origins of anti-CRISPR proteins, which Kevin has actually been working on really nicely. So I just want to thank you for your attention, for keeping up with this virtual seminar. Special thanks to Michael Emmerman, who's really actually a virologist instead of me being a pretend virologist. This entire line of work actually started because of influential coffee I had with him almost 17 years ago when I started my lab. And actually 60% of both of our lab's productivity is the direct result of a collaboration that really stemmed from that coffee conversation, which I'm really grateful to him for. I also want to thank Patrick and Rosanna, who did a lot of the work. Also Emily and Rachel, who did this work in my lab. And also our collaborators at the University of Freiburg. And then the TRIM5 work has actually been completely led by Jeanette Tenture and aided by two remarkable young scientists who are all going to be applying to grad school. Candace actually just joined grad school at the University of Washington. So thank you for your attention. And I will stop sharing and open myself up for questions. I think I'll transfer host duties back to Larissa, if that's okay. Hopefully there are questions. I'm happy to take them. Or Malik, you're happy to read them out to me.
Yes, thank you, Harmit. There are questions, and I'll go ahead and it looks like there are some questions in the chat as well as in the Q&A. So I'll start off with a question here that says, is there a difference between the key residues amongst MXA splicing variants? that would make sense since different splicing variants can be expressed in different cell types. And different cell types are susceptible to different viruses. So do you have any comments on that, Harmit?
Yeah, that's an excellent question. So there are at least 2 predominant splicing variants in MXA, but actually none of the positively selected sites are retained in one and not in the other. They're basically both retained. I think the splicing variants mostly affect the subcellular localization of the MXAs being produced. There's a little bit of controversy about the effect of the second splicing variant. What I showed you is actually the predominant version, but in some cells, a shorter version of MXA is actually produced. And some people have argued that that is actually induced by viruses such as CMV to make a dominant negative version of MXA, but that actually has not been reproduced. So there's no evidence that the second splicing variant has different antiviral specificities. But for other antiviral proteins, not for MXA, that certainly appears to be a possibility that we have explored and actually published on in one case. So splicing variation is definitely an additional means for host cells, apart from the fact that they are diploid and have allelic variants. On top of that, splicing variation is a great way for the host to actually graft even more variation onto its genome.
Thank you. Here's another question. She says, fantastic talk, Harmit. This question is from Antonis. It says, can you comment on what are the levels of variation in the L4 loop in MXA in the human population?
Yeah, excellent question. I actually get that question almost every time. I should put a slide. Actually, human MXA is remarkably monomorphic at the L4 loop. As actually monomorphic for most of MXA. There is one variant that is actually super interesting to us that is at about 5% frequency across different kind of ethnic populations, but it does not coincide with any of the positively selected variants, and people have extensively examined that for differences in antiviral activity. That variant meets the criteria of what we would call over-dominant or balancing selection, but we do not know what is actually driving that. It is not influenza-like viruses or other RNA viruses that we've actually tested. But there's no variation in loop L4 in humans. Now, that's a different story in old world monkeys, where we do see variation, including in African green monkey and rhesus, which we are actually exploring what the functional consequences of that might be.
Thank you. Here's a question from a trainee. And here's one actually I have not considered before. So it says, since HIV causes a slow disease, can we still use the same assumptions about host virus armorages for it? Considering the short life expectancy for most of the human history, would it even be an important factor in selection?
Oh, that's an excellent question. We actually don't think that HIV is the reason why TRIM5 has actually evolved the way it has, at least among hominoids. However, SIV-like viruses are at least 20 million years old now, based on our kind of and others' analysis among old world monkeys, and so easily could actually be an influence there. However, what I didn't actually talk about very much here, but is something that we've published on before, is that the predominant specificity for human TRIM5 has actually been driven by MLV-like viruses that are actually part of your genome. So these are endogenous retroviruses that actually do create a fitness detriment, but they don't do so in an infectious fashion. Instead have actually been a cumulative fitness detriment that they've caused. So we use HIV almost as a tester virus, just like we use Tagotovirus as a tester virus to actually look at the phenotypic consequences of MXA. But our ability to actually make a strong statement about which virus likely led to the particular change that we have assessed in extant species is extremely poor, because our ability to look backwards in evolution for host genes is very high, but with viruses, it's extremely shallow. So we are basically making lots of, actually no assumptions about which viruses actually likely drove this, except it likely was a very similar virus to the one that we are actually using in the lab.
Thank you. Here's a question from a trainee. I don't know if I necessarily understand it, so I'll just let you interpret it.
Yeah, is it in the Q&A or in the
It's in the Q&A, but I'm happy to read it out because it's a small one. It says, I was wondering, have you thought about how TRIM5 activity behaves at the systems level? And could this explain the lack of antiviral activity in humans?
Yeah, I think I understand what the question is. So I think the question alludes to the fact that even though TRIM5 alpha, everything I presented to you is in the context of an antiviral state against HIV, TRIM5 alpha also has a function in terms of potentiating an NF-kappa B-like immune response, and having an overactive version of TRIM5 alpha would basically give you an autoimmune-like effect. So I didn't tell you about what TRIM5 alpha is. It's a E3 ubiquitin ligase that actually modifies proteins and targets them for proteasomal degradation. And you can imagine that if you actually had a very rapidly evolving specificity in E3 ubiquitin ligase, at some frequency, you would probably hit some host protein that would be detrimental. We don't think that that would be true because, of course, all our assays are done in the context of human cells. However, the potentiation of immunity is definitely a possibility. And we are always making this trade-off between having a hyperactive immune response in the presence of a pathogen, but a very low response in the absence of a pathogen to prevent autoimmunity. And it's possible that some of these arginine residues that we've chosen to fix in our species as a result of that dampening autoimmunity effect.
Thank you. Here's a question that I often sort of wondered about when I was working on some host-pathogen interaction questions. It says, how strong this restrictive effect of a protein should be to prevent infection? In your experiments, were the proteins expressed with a native promoter or were they overexpressed? In other words, how much inference can you make regarding the ability of the protein to actually prevent infection based on the restrictive effects?
Yeah, so again, an excellent question. So the way we do our experiments, we are sort of downstream of where people have actually identified these viruses and these antiviral effects in the context of a native knockout. So that's actually how MXA's effect against influenza, like I talked about, where you knock out MXA or put back MXA under an endogenous promoter and show virus infections. Once we do that, then it's a function of actually maximizing your chances of actually seeing what the dynamic range of the assay is. So we actually use different promoters depending on what type of restriction we are looking at. If it's a very potent restriction, we actually lower the promoter activity of what we're looking at because, so for instance, MXA is actually induced 10,000-fold within 10 minutes of interferon. Now that would blow out any assay we do. So that's a case where we actually downregulate the amount of MXA that we're producing because we want the dynamic range of our assay. So all of these assays are motivated by things that were made under native context, but the nature of the assay and how we basically assess them in cell lines is really motivated by maximizing the dynamic range so we can actually look for the effects of single changes. So it's a good question, but doing these experiments in the context of native or endogenous promoters often is actually not going to give you the answer that you're looking for.
Yeah, that makes sense. Thank you. Final question here from Seth. It says, maybe I missed this, but if one can, by example, or for example, mutate 10 positively selected, 10 neutral, and 10 negatively selected sites in the same area of the MXA structure, would these mutations in these three L3 categories show general differences in antiviral breadth? In other words, are there rules across these three categories of evolution?
Yeah, that's an excellent question. Actually, I was running out of time, Seth, so I didn't actually make this point. Based on what I told you in terms of first principles, I would have suggested that because V1 loop has both completely conserved residues and positively selected residues, I would have predicted that actually mutations in conserved residues would have actually very little impact, and mutations in very positively selected residues would have a bigger impact. That is actually not what we see. However, it seems that mutations outside V1 loop, which are in conserved positions, are more likely to be detrimental for function, and mutations that are not very well conserved are likely to be intermediate. So it seems that the entire V1 loop, just like the entire loop L4, which has actually got a high propensity of these positively selected sites, is actually affecting the functional impact even of the neighboring sites. So even though we would have predicted that absolutely conserved residues would be exceedingly recalcitrant to mutation, in our assays they were not. In fact, mutations in that glycine residue that is completely conserved in primates was mostly beneficial for HIV restriction. So that is definitely a surprise of our assay. We don't actually have a lot of evidence to say things beyond the V1 loop, because that's the one that we've actually mutated in depth. But as we go forth, I think we will find more instances where we can actually look for differences between neutrally evolving, well, nothing's really neutrally evolving, but things that are under mild purifying selection, strong purifying selection versus positive selection.
Thank you. Perhaps I'll ask the last question here. So do you have any ideas as to why the human TRIM5 is so resilient against the MLV? Is it because it is using multiple surfaces, and so it doesn't really matter if you disrupt one of the surfaces, or is there some other hypothesis?
Yeah, there's actually two hypotheses here. One hypothesis is that it's actually the height or the fitness peak. So human TRIM5 alpha is really, really good against MLV. It takes very little human TRIM5 alpha to restrict entropic MLV. So perhaps it takes multiple mutations to knock it off the fitness peaks. That's one hypothesis. And so any single mutation is very unlikely, although we did find a few, but any single mutation is very unlikely to knock it off the fitness peak. That's possibility number one. Possibility number two is actually that just like with HIV, if this is primarily driven by electrostatic charge, again, it takes multiple changes to actually manipulate that property in a significant way to lose the activity. The third possibility is actually what you pointed out, is that because of the avidity, which is really the predominant determinant, individual affinity between monomers can actually take quite a significant hit without actually affecting the avidity of the interaction. That is, I suspect, actually that's Jeanette's favorite hypothesis, and maybe it was designed to do so to actually prevent significant amounts of viral escape, because actually you can increase avidity by continue to increase the TRIM5 interaction. And so you're actually less dependent on the TRIM5 capsid interaction. Even a small change there can actually lead to a significant boost. And I suspect that's why a lot of these antiviral changes, antiviral proteins act in this oligomeric fashion where they can translate small adaptive value changes to a pretty big adaptive effect.
Great, thank you. So that wraps up the Q&A. Harmit, thank you again so much for coming to give a seminar. So I believe there is a reception now to which anyone, I believe, is welcome. I believe you have to leave the Zoom and come back, but perhaps Larissa can give a little bit more instructions as to what the attendees are supposed to do. Should they hang around or do they also need to log off? Larissa, could you please comment?
No problem. Can you hear me?
Yes.
Okay, perfect. Yes, so if everyone can log off, and then I just sent the Zoom link around. It's also in all of the emails that have been going out. If you just click on that, it'll be a traditional Zoom where everyone can see each other. Thank you so much.
And Larissa, would you mind actually copying and pasting all the questions in the chat and the Q&A? And if I didn't answer any in real time, I'll try to send answers through you or through Malik.
Absolutely, sounds great, thank you.
Thank you. Thank you, guys. See you in a bit.